Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Axoneme. 20 languages. Axoneme. Eukaryotic flagellum. 1 axoneme, 2 cell. Membrane, 3 IFT, intraflagellar transport, 4. Basal body, 5 cross-section of flagellum, 6. Triplets of microtubules of basal body. Cross-section of an axoneme in a flagellum. This article includes a list of general references, but it lacks sufficient corresponding inline citations. Please help to improve this article by introducing more precise citations. May 2009, learn how and when to remove this message. In molecular biology, an axoneme, also called an axial filament, is the microtubule-based cytoskeletal structure that forms the core of a cilium or flagellum. 1, 2. Cilia and flagella are found on many cells, organisms, and microorganisms, to provide motility. The axoneme serves as the skeleton of these organelles, both giving support to the structure and, in some cases, the ability to bend. Though distinctions of function and length may be made between cilia and flagella, the internal structure of the axoneme is common to both. Structure, edit. Inside a cilium and a flagellum is a microtubule-based cytoskeleton called the axoneme. The axoneme of a primary cilium typically has a ring of 9 outer microtubule doublets, called a 9+0 axoneme, and the axoneme of a motile cilium has 2 central microtubules in addition to the 9 outer doublets, called a 9+2 axoneme. The axonemal cytoskeleton acts as a scaffolding for various protein complexes and provides binding sites for molecular motor proteins such as kinsin II, that help carry proteins up and down the microtubules. 3. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF identifiers. Mesh D054468 THH1.00.01.1.01017. Anatomical terminology. Edit on Wikidata. Micrograph of thin cross section of chlamydomonas. Axoneme. A simplified model of intraflagellar transport. Primary cilia, edit. The axoneme structure in non-motile primary cilia is of an outer 9 microtubule doublets with no central microtubule singlets, and no dynein arms on the outer doublets. This arrangement is known as the 9+0 axoneme. Primary cilia appear to serve sensory functions. Motile cilia, edit. The building block of the axoneme is the microtubule. Each axoneme is composed of several microtubules aligned in a characteristic pattern known as the 9+2 axoneme, as shown in the image at right. 9 sets of doublet microtubules, a specialized structure consisting of 2 linked microtubules, form a ring around a central pair of single microtubules. Besides the microtubules, the axoneme contains many proteins and protein complexes necessary for its function. The dynein arms, for example, are motor complexes that produce the force needed for bending. Each dynein arm is anchored to a doublet microtubule by walking along an adjacent microtubule. The Dynein motors can cause the microtubules to slide against each other. When this is carried out in a synchronized fashion, with the microtubules on one side of the axoneme being pulled down and those on the other side pulled up the axoneme as a whole can bend back and forth. This process is responsible for ciliary forward/flagellar beating, as in the well-known example of the human sperm. The radial spoke is another protein complex of the axoneme, thought to be important in regulating the motion of the axoneme. This T-shape complex projects from each set of outer doublets toward the central microtubules. The interdoublet connections between adjacent microtubule pairs are termed Nexin linkages. History of discovery, edit. The first investigation of sperm flagellar morphology was begun in 1888, by German cytologist Ballowitz, who observed using light microscopy and mordant stains that a rooster sperm flagellum could be splayed into as many as 11 longitudinal fibrils. About 60 years later, Grigg and Hodge in 1949 and a year later Manton and Clark observed these 11 fibers in splayed flagella by electron microscopy. These investigators proposed that 2 thinner fibers were surrounded by 9. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Thicker outer fibers. In 1952, using advancements in fixation, embedding, and ultramicrotomy, Fawcett and Porter proved by thin sections that the core of epithelial cilia within the ciliary membrane consisted of 9 doublet microtubules surrounding 2 central, singlet microtubules, i.e., the central pair microtubule apparatus, and hence the term, the 9+2 axoneme. Because of the high degree of evolutionary conservation between cilia and flagella from most species, our understanding of sperm flagella has been aided by studies of both organelles and from species ranging from protists to mammals. Cilia are typically short, 5-10 m, and beat in an oar-like fashion, with an effective stroke followed by a recovery stroke. Flagella beat with a snake-like motion and are typically longer, generally 51-50 m, but ranging from 12 micrometers to several millimeter in some species, with flagellar length in the protist chlamydomonas being regulated by several genes encoding kinases. It was recognized first by Manton and Clark that the 9+2 axoneme was possibly ubiquitous among species, and indeed, the 9 doublet microtubules are evolutionary conserved structures that evolved in early eukaryotes nearly 1 billion years ago. However, there is wide variation among species with regard to the detailed structure of sperm flagella and their accessory structures. Axonemal doublet microtubules assemble from the ends of 9 centriolar forward/basal body. Triplet microtubules, whose 9-fold symmetry and clockwise pinwheel pattern, looking from inside the cell to the flagellar tip, is organized by the conserved protein of the Sas6 gene, and which is introduced into some eggs to establish the first mitotic spindle. The 9 doublet microtubules are then connected around the axoneme by Nexin links. Currently, the molecular structure of the axoneme is known to an extraordinary resolution of 4 nm through the use of cryo-electron tomography, as initially pioneered by Nicastro. Sperm flagella and ciliary motility has been effectively analyzed in simple systems, for example, protist flagella and sea urchin sperm, whose flagella contain several hundred polypeptides by proteomic analysis. For clinical significance, edit. Mutations or defects in primary cilia have been found to play a role in human diseases. These ciliopathies include polycystic kidney disease, PKD, retinitis pigmentosa, Bardet-Biedl syndrome, and other developmental defects. References, edit. 1 axial filament. thefreedictionary.com. Retrieved 9 May, 2021. 2 Porter Me, Sale WS, November, 2000. The 9+2 axoneme anchors multiple inner arm dyneins and a network of kinases and phosphatases that control motility. The Journal of Cell Biology. 151, 5. F37-42. Do. 10.1083/JCB.151.5.F37. PMC 2174360. PMID 1108617. 3 Gardner MB, September, 2005. The Importance of Being Cilia. HHMI Bulletin. 18, 2. Archived. From the original, PDF, on the 11th of March, 2010. Retrieved the 18th of March, 2010. 4 link, Richard W. Keams, Hector, Albertini, David F. February, 2016. The Axoneme, the propulsive engine of spermatozoan cilia and associated ciliopathies leading to infertility. Journal. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Of Assisted Reproduction and Genetics. 33, 21411562, 10.1007/016-0652-1. ISSN 1058-0468. PMC 47590005. PMID 268258807. Material was copied from this source, which is available under a Creative Commons Attribution 4.0 International License. Further reading, edit. Wilson C.W., Nguyen C.T., Chen M.H., Yang J.H., Gaikian R., Huang J., Chen J.N., Chuang P.T., May 2009. Fused has evolved divergent roles in vertebrate hedgehog signaling and motile ciliogenesis, PDF. Nature. 459, 7243 98102. Bibcode, 2009 Nature.459, 98W. Do. 10.1038/Nature 07883. PMC 32048898. PMID 193053993. Fogel G., October, 2005. News Focus, Betting on Cilia. Science. 310, 5746-2168. Do. 10.1126/310.5746.216. PMID 162239997. S2 SID 8343337. Porter Me, Sale WS, November, 2000. The 9+2 axoneme anchors multiple inner arm dyneins and a network of kinases and phosphatases that control motility. The Journal of Cell Biology. 151, 5 F37-42. Do. 10.1083/JCB.151.5.F37. PMC 217430. PMID 11086017. Dylan R.H., Faus ILJ, December 2000. An integrative model of internal axoneme mechanics and external fluid dynamics in ciliary beating. Journal of Theoretical Biology. 207, 341530. Cite CX 10.1.1.127.4124. Do 10.1006/b.2000.2182. PMID 11082310. Omoto CK, Gibbons IR, Kamiya R, Shingi OGC, Takahashi K, Whitman GB, January 1999. Rotation of the central pair microtubules in eukaryotic flagella. Molecular biology of the cell. 10.114. Do 10.1091/MBC.10.1.1. PMC 25148. PMID 988031. Rosenbaum J.L., Cole D.G., D.N. D.R., February 1999. Intraflagellar transport, the eyes have it. The Journal of Cell Biology. 144, 33858. 10.1083/JCB.144.3.385. PMC 213291. PMID 9971734. Categories: Eukaryotic cell anatomy organelles. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.